mentioned, and we've been investigating the influence of microplastic color and odor on feeding preferences in Daphnia magna. And you can see a really beautiful video of the Daphnia under a microscope here. So they're freshwater crustaceans, and they're quite small. They're about the size of a grain of rice. You can see some Daphnia feeding on some microplastics here in one of our early experiments. So they are quite small, not quite microscopic, but they were the model organism that we chose to work with throughout this project. And the other key part of that title was microplastics. So what are microplastics? Well, they're small pieces of plastic. They're normally less than 5 millimeters. We see primary microplastics, and those are the kinds that are manufactured to already be at that small size. So items like glitter or exfoliating beads that are sometimes found in cosmetic products and toothpastes. And we also have secondary microplastics. And these are the kind that come as plastic doesn't biodegrade, but instead just breaks down by fragmenting into smaller and smaller pieces over time. And once those pieces reach 5 millimeters or less, they're then classified as microplastics. At this point, microplastics are everywhere. We see them in the ocean, they're in the soil, they're on the top of mountains, they're in our drinking water, in the air we breathe, even dust in our homes. And they are incredibly prevalent in our environment. And any nook and cranny that microplastics could possibly have gotten themselves into, they are there. But when we look at aquatic ecosystems and the ocean, we're seeing that the most common type of microplastics here are synthetic textiles. And those are the kind that we worked with for our project. So if you own polyester clothing at home, when you wash those clothes in the washing machine, they shed microfibers, which are a type of microplastic. And then those microfibers end up being washed out into the water system and eventually make their way into fresh water and the ocean. And once they get into those ecosystems, we're seeing that they're being ingested by small aquatic organisms. But the story doesn't quite end there, because there are bigger concerns of bioaccumulation and biomagnification. And this is the idea that when these tiny organisms are eating these microplastics, well, something eats them, and then something eats that, and then something eats that. And so these microplastics end up moving up through the food chain. They don't just disappear after they're ingested by these small aquatic organisms. And so as they move up the food chain, they're accumulating in these higher level predators. And we have similar concerns with biomagnification. And this is the idea that these plastics are produced with some chemicals. They're also absorbing different chemicals and toxins during their time in the water. And that those chemicals and toxins can become potentially more potent and more harmful as they accumulate in these higher level predators, which includes us. So microplastics are really prevalent in our environment, and it's important that scientists begin to understand what aquatic organisms are eating them and how they're moving up in the food chain and potentially affecting humans as well. So we knew we wanted to work with microplastics to focus on microplastics as part of this plastic crisis that we're dealing with, but then we needed to figure out how are we going to actually analyze these fibers. So what you can see here, there's a little swatch of polyester fabric from Joann's. And I cut off the top fluffy layer of that fabric. And then I cut it up super, super fine, in this case with a scalpel and later with razor blades as we were working with bigger batches, and essentially made some homemade microplastics. And that's what you see on this slide here. And then we were able to take those microplastics and take a look at them under a microscope. And what you see here, we have this column of bright field images. And our initial concern was that we were going to have some trouble identifying what color these microplastics were underneath the microscope, especially once they were in the intestines of these Daphnia. And so you can see that these colors, especially for green and blue, tend to get a little bit washed out, and they're a little harder to identify here. But we found that these plastics actually have autofluorescent characteristics. And so we saw that these red plastics here actually emit in the red emission. And then the key here was that these green microplastics emit in red as well, but they also emit in this green emission here. And this was really important because we were able to analyze these green microfibers using this green emission, this autofluorescent characteristic here. And then we have our blue microfibers, which also emit in red, the key being that they didn't emit in the green, and then they emit really, really faintly in blue. But because these green microplastics emit in this green emission here, we were able to use this autofluorescent characteristic in order to identify those plastics when they're in the intestines of our Daphnia. And at the bottom, we see our black microfibers. And because the black is so dark originally, they were easy enough to see just using those bright field images. So we're able to use these autofluorescent characteristics It was instrumental in us being able to go through and analyze these fibers and get the data that we needed after these Daphnia had ingested the fibers. So then we were able to move into experimentation. I wasn't quite done with microplastic preparation yet. I also had to go through and make some bigger batches that we could actually use when it came time to add these microplastics into our experimental jars. And so what I did was I created some big batches of each of the colors that we were working with. And then we were able to weigh those out and add them into some combo media and create what we called stock solutions, which you see here on the left. And so we were able to have these pre-made stock solutions where we knew the concentration of microplastics. So it was a really efficient and easy way to have these microplastics prepared and ready to go. So we could just pipette them into our treatment jars when the time came to start an experiment. In addition to that, we also had some Daphnia culture throughout the semester. And this allowed us to remove some adult Daphnia when we were ready to conduct an experiment. And then we could fast them for 24 hours. And that was to ensure that the Daphnia were hungry, that they would eat these microplastics, and also to make sure that their guts weren't too opaque, that they were semi-transparent, and we had as much visibility as possible when it came to looking through the intestines and seeing these microplastics. And then we had two main sets of experiments here. So we did a series of color experiments, and then independent of that, we did a series of odor experiments. And in both cases, we exposed the Daphnia to these microplastics after they were fasted for those 24 hours. And then we were able to go through and fix the animals, which you can see in process here on the right. Then those intestines were dissected out and imaged, and then we could go through and analyze that data, again, relying on those autofluorescent characteristics that I talked about before. So as we moved into our first set of experiments, we were looking at color preference here. And my initial hypothesis was that Daphnia would have a preference for green microplastics. And this is because I thought that those green microplastics would mimic and closely resemble algae, which is the normal food source for Daphnia. And when I was researching some of the other aquatic animals that are commonly eating plastics, they tend to be eating plastics that look most similar to their normal food source. So the example that you might be most familiar with is that sea turtles were reported for eating plastic bags in the ocean. Why? Well, because to a sea turtle, those plastic bags look an awful lot like a jellyfish. So the thought here is that these Daphnia would mistake these green microplastics for algae and be more drawn to them and eat more of them. So when we came to do our 3 treatments, we did a 50-50 mix of green microplastics mixed with either black, blue, or red. And for all of these treatments, again, we fasted those animals for 24 hours. And then those 8 animals for each of our groups were exposed to a final concentration of 0.01 milligrams per milliliter of microplastics for 1 hour. And you can see this was one of our groups here. You can see those 8 Daphnia. Some of them are currently feeding on some microplastics at the bottom here. And then after that hour was up, they were removed from these treatment jars, fixed. The intestines were dissected away. We could image them, and then we could go through and analyze that data. And so what we saw is that when we took a look at these intestines, you can see the bright field images at the top here for our black and green group, our blue and green group in the middle, and then our red and green at the end. That those bright field images, it can be a little bit hard to tell and distinguish those different fiber colors, especially for this blue and green group here. The red's a little bit easier to see, it stands out a bit more. But for our blue and green, most definitely, that's where having these different color emission spectra came in super handy. So this red emission spectra in the middle here, that's showing all of the color fibers except for black, as all of the fibers had those autofluorescent properties in red. But the key here was these green emission spectra at the bottom. And so these photos here are just showing those green microplastics. And so this allowed us to go through using these green emission photos here to count the number of just green microplastics. And then we could refer back up top to our bright field images to count the total number of plastics that we were seeing in a representative area of the intestine for each of these animals. And then we could subtract the number of green from the total number that we're seeing here to make sure with a high degree of confidence we were accurately counting and identifying these fibers. So we went through and we did that for the 8 animals in each of these groups. And when we took a look at our data, what we were seeing is a slight preference here when you look at the percent of microplastics for black and for blue, and then a slight preference in our red and green group for green here. If you take a look at that data presented as the ratio of ingested microplastics here, So 1 being a 1-to-1 ratio, where you had 50% of green and then mixed with the other color in the intestine. Again, we're seeing just above 1 for our black and for our blue. So that slight preference for black and blue plastics here. And then just under 1 representing that slight preference here for green. But across the board, these results were not statistically significant. So what we were able to conclude from this initial set of testing is that microplastic color alone doesn't influence preference for these plastics. But things got a little bit more interesting when we moved into our odor experiment. So here we had a focus on looking at the odor, the olfactory cues that could be secreted by these microplastics. And my initial hypothesis was that the Daphnia would have a preference for biofouled microplastic. And what that means is that we took some microplastic and we put them in combo media, and then we added some algae. And that's this 2 tubes back here. So there's black and green, which are the 2 colors that we chose for the set of experiments. And they were both being aged with algae in our greenhouse for about 3 weeks. And then up front here, we also have black and green fibers, and they were aged without algae. So these are our pristine microplastics in the front here. And then our biofouled or plastics aged with algae are in the back. And what we were able to do is then recombine these different samples to create our 4 treatment groups. We have pristine green microplastics mixed with either pristine or foul black, and then foul green microplastics mixed with either pristine or foul black. And the assumption would be that these Daphnia will prefer the microplastics that have been biofouled because they would be able to mimic those olfactory cues that are normally secreted by algae and therefore seem more attractive to the Daphnia. So again, we had 8 specimens for each of these groups. They were all fasted for 24 hours, and then they were exposed to this final concentration of 0.1 milligrams per milliliter of microplastics for 1 hour. And in this case, we did one initial set of testing before we took a look at the intestines of these Daphnia. So for each of these tubes here, we took a sample and we stained it with some Daffy. And Daffy stains nucleic acids, which is the building block of DNA, as you can probably guess, blue. And so what you're seeing here is we have our pristine microplastics at the top. So these are the ones aged without algae. And when we took a look at the Daffy staining, we're seeing some minimal staining here, and that's probably just from some bacteria growth. And when we say these microplastics are pristine, we're simply referring to the fact that there was no algae present during the aging process, not that they were necessarily sterile. There was some potential bacteria growth here, which was fine. But more importantly, in our biofoul group, you can see this, what almost looks like a little splotch or a smudge here. And that's actually a cluster of algae, which you can very clearly see in this Daffy staining group here. And so that confirmed that we had algae growth in our group where we were aging these microplastics with algae, which is what we wanted. So that was great. And that we weren't seeing any algae, so there was no contamination in our pristine group. Also great. So with that in mind, we're able to go forward and take a look at the intestines. And again, here's one representative animal for each of these groups. So we have our pristine microplastic group with the green and the black here first. Then we have some foul green and some pristine black microplastics, some pristine green and foul black. And then down at the end here, we have our foul green and our foul black microplastics. And we use the same technique here for counting. So we have our green emission images down at the bottom. And these are used so we could go through and count all of these individual green fibers here for each of our animals. And then we could go back and take a look at our bright field images to count all of the fibers that we saw. And we still did it this way, even though the black microfibers were a little bit easier to see, because there are some sections of the intestine, like this one here in this first photo, that were a little bit more cloudy, a little bit harder to see. And we wanted to make sure that our counts were accurate. And so by using this green emission imaging here, we knew with a high degree of confidence that we were just counting these green microplastics. Then we could go back, get the total, subtract the number of green to have an accurate count of black without any potential for misidentifying these fibers. So after we went through, counted all of these fibers for all of our animals, we took a look at that data, and we're looking at the percent of microplastics here. We're seeing a clear preference across the board for black, which is very interesting. So my initial hypothesis was Daphnia would prefer green microplastics, but that wasn't the case here. And what was more important is that when we had microplastics that were biofouled, those microplastics that were aged with algae, we're seeing a statistically significant preference here for these black fibers. And that was really important. And when we take a look again at the same data, but presented for the ratio of ingested microplastics, with one being a one-to-one ratio of black to green. Again, across the groups, we're seeing just above one. So just a teeny bit above one here, so a slight preference for pristine microplastics. You see that preference is increased. Now it's statistically significant. We had half of our fibers aged with algae. And you can see very clearly here a really intense and heightened preference for black microplastics. We had fouled green and fouled black microplastics here. And so our working hypothesis and the thought is that Daphnia are eating more black microplastics. We're seeing this preference here because those microplastics that were aged with algae are able to absorb those chemicals that algae normally release. And then when we add it and mimic what algae normally does, and by releasing those olfactory cues into the water, it was encouraging feeding behavior in the Daphnia. And so there's this underlying preference for black microplastics. But when Daphnia are in the presence of these olfactory cues, it's encouraging this intense feeding behavior. That preference for black microplastics is really heightened and intensified and becomes noticeable. And the thought is that it's simply these olfactory cues that are in the water that are causing this heightened preference, as opposed to the individual fibers themselves. Because if we look here at this group, where we have fouled green microplastics and pristine black, if it was the individual fibers that were being more attractive to the Daphnia, we would be seeing a preference for green here because those are the microplastics that were biofouled. But that's not the case. We're still seeing this preference for black. So that's leading us to believe that it's these olfactory cues in the water encouraging this feeding behavior that are heightening this underlying preference for black microplastics that was already present, but otherwise goes unnoticed unless you have this really intensive feeding pattern happening. So that was our main takeaway here from this set of experiments. And if we go back and we recap some of our key findings, all the way back in the beginning, outside of our results, the fact that these microplastics can be identified by these unique emission spectra when we looked using the fluorescence microscope was really important. It was instrumental in us being able to identify these fibers and go through and analyze our results. And this is something that could in theory be used for other experiments and other research as well. Being able to go and use this emission spectra and these unique autofluorescent properties of these plastics enable scientists to better count and more accurately count different colored fibers if they're looking at fiber color preference inside of different animals. In terms of our actual results, so we saw that this microplastic color is not a significant factor in fiber preference. But when we have these olfactory cues present, now we see this preference for black. So these Daphnia have a preference for black microplastics when you have these olfactory cues present that are normally secreted by algae, but in this case are being secreted by these biofouled microplastics. And that was our main takeaway over the course of this year. And if we had more time, we would love to continue doing further experimentation to confirm that these olfactory cues are in fact diffusing into the water from these microplastics. And that would be really great to ensure that our hypothesis is correct, that these microplastics are in fact secreting these chemicals into the water, and that's causing that encouraged feeding behavior and that heightened preference for black microplastics. We would also like to be able to determine the threshold for these olfactory cues to see that impact and preference. So we saw when half of our microplastics were biofouled with algae, there is a somewhat increase in this preference for black microplastics compared to where both groups, the black and the green, were biofouled microplastics and algae. Now we're seeing this really, really intense heightened preference for black. So to be able to take a look and determine those thresholds for these olfactory cues would also be a great extension of this project. Those interested in chemistry, taking a closer look and identifying what chemicals are actually being secreted into the water by algae, and then by extension being absorbed and then secreted by these microplastics would also be a really great extension. And lastly, we would love to be able to determine if these findings are true for other types of microplastic. So here we were using microfibers from polyester fabric, but different types of plastic vary in their ability to absorb and secrete different chemicals. And so it'd be interesting to see if other types of plastic are also able to act the same way and cause that increased heightened preference for black microplastics when they're aged with algae. But unfortunately, the year is coming to an end and we don't have any more time. But I would like to thank Dr. Christensen for working with me really closely on this project, taking this project on, and all of his help when it came to troubleshooting as we had some bumps along the road, and all of his help with going through, analyzing all of this data. I'd also like to thank Dr. Stevens and Dr. Giuliano. They're part of my honor committee, and their advice and feedback throughout this process has been really beneficial. And I'd like to thank the biology department team as well. I feel quite fortunate to have the opportunity to do independent research here and have access to all of these great resources and facilities. And so it's been a really great experience to be able to work on this project, do some independent research, and have some lab experience. And of course, thank you all for listening today and learning a little bit about the world of microplastics and Daphnia with me. And if anyone has any questions, happy to answer them. All right, hold on. Okay, so I can see everybody.
I have a question, Jean.
Oh, okay. Either one. Dr. Christensen raised his hand as well.
Oh, okay. He can go first.
Oh, thank you. Great job. That was awesome. I really appreciate that. I actually have two questions. One is more general and then one's more specific about your research. But I'm curious, when the plastics, so they bioaccumulate and then they biomagnify up the food web. Do they persist in the gut and the intestines, or do they break down some more and then get incorporated elementally into the tissues? Do you know, or do they just get excreted outward?
Yeah, so it depends. There are some microplastics that are able to just be excreted, but some do end up accumulating in the tissue. And I know there are some ongoing studies, especially looking at seafood. And there are concerns that the seafood that we're eating, that these microplastics might be in the tissues of some of these fish, and that could potentially then be impacting us. So there are concerns that these microplastics aren't always able to be excreted and passed, and that can cause these larger problems when you're looking at bioaccumulation and biomagnification across the
wow.
web.
My next question, and I'm curious, did you happen to look at, and I would imagine this is really hard to do, but did you happen to look at size of the microplastics in relationship to consumption?
Yes, well, we looked at the general size overall. I don't think we did any statistical testing for looking at what size is preferred. But for the most part, it seemed that the smaller the microplastics were, like as small as I could get them, those were the ones that were most often eaten compared to some of the comparatively longer pieces. So it seemed like smaller microplastics were preferred overall, but I don't have data to confirm that for sure. But
thank you. Good job.
You're welcome. Thank you.
Okay, Jean, my question for you is, when you were growing your algae in your black and your green, kind of making your biofouled specimens, was there a way to ensure that the algae kind of grew and affected the green and black microplastics equally, or was there perhaps more growing in one than the other?
We did go through, we did some counts throughout the process. We had some contamination issues the first time we did this. So the second time we were a little more cautious and we did consistent counts of algae in those two samples. And I think they were pretty close to equal. So based off of just algae counts and growth, I think they were pretty close to the same, but I can't speak to how they're impacting individual fibers. But the hope was that we treated them the same. They were in the greenhouse for the same amount of time, that hopefully they were impacted the same way by algae for both of those groups.
Okay, thank you.
Hi, Dr. Berman.
Hello, great presentation. Thank you for showing your data sharing your data with us. I just have a quick question, and I'm sorry if you mentioned this. Under normal circumstances, what are the cues that bring Daphnia to their food? Is it only olfactory, or are there other cues that help them navigate to food?
I'm not an expert about feeding patterns on Daphnia, unfortunately, so I'm not entirely sure. I do know that they're traditionally filter feeders, and they will move through the water column looking for food. So my guess would be that I know the olfactory cues definitely seem to play a role, but aside from that, I'm not quite sure about other cues, what else Daphnia are looking for.
And do they, that's a dumb question. They don't have eyes, right? They can't see.
So they do. They
Oh,
have an
do.
eye.
Oh.
Yeah.
Okay.
And so
Okay.
there was experiments done. They can see different colors, which was important. That was part of the
Right.
foundation.
Yeah, right.
To
Okay.
make sure that they could see the different colors. So there was a reason to test for color preference there.
I'm just wondering, like, in addition to the olfactory cues, are there other things that, some type of like chemotaxis or something that brings the Daphnia to the food?
Yeah, I know
Anyway.
that they're sensitive to chemicals secreted in the water, especially for things like predators. They're very aware of like chemical cues in the water, but I don't have a ton of background about what chemical cues specifically are being secreted by the algae or just in general other things that are attracting them to food, unfortunately. Sorry.
Because I think that last question they had is really interesting. Like, what is it about what's in the algae that's kind of drawing them to certain microplastics? I think that's a really interesting, I mean, I know you're graduating, so, but that would be an interesting next question.
If Dr. Christensen, he has a little more background on Daphnia, and I was a little bit more into the plastics and environmental side of it, so maybe he can help answer.
Yeah, they're definitely very sensitive to chemical cues in the water. So there's like amazing work that's done with some of their predators and even the water that their predators live in, applying that water to the media that Daphnia are in. The Daphnia will change their shape so they can't be caught by the predators. And there's also epigenetic heritability there where their offspring will have these bizarre shapes that allow them to avoid predation. So in general, super sensitive to chemicals.
Yeah.
Some of the VOCs that algae secrete, there's more work that actually shows that they reduce feeding behavior. So that it's like the algae have a protective way to avoid being consumed.
Oh, wow.
A lot about attraction, and the running hypothesis here was that if these microplastics are grown with algae and some of those VOCs are absorbed, not necessarily the repelling ones, but certainly there must be some types of chemicals that actually the Daphnia are attracted to, but there's not a lot of work with that. So that gets to Jean's interest in maybe doing some analyses of the water and seeing what types of chemicals there are.
Mm-hmm. Yeah, maybe some chemistry. Yeah, that's really interesting. Thank you so much.
Yeah, yeah, great job. Yeah, it's very interesting topic and research and great presentation. So